I'd like to introduce Lindsay Pease. She is an assistant professor and extension specialist in nutrient and water management at the University of Minnesota Northwest Research and Outreach Center in Crookston. Lindsay's work focuses primarily on nitrogen and phosphorus management in row crop systems with an emphasis on nutrient loss and agricultural runoff. Lindsay attended the Ohio State University, where she received her PhD, MS, and BS in food, agriculture, and biological engineering. Lindsay?
All right, thank you. And I noticed Tom did get the Ohio State University in there. Thank you. No, it's always funny to actually kind of say that out loud. But yeah, but I've been here in Minnesota for about 5 years, and yeah, so we're just getting into some of this nutrient management stuff. As Tom mentioned, I am up at the Northwest Research and Outreach Center, so following Matt Rourke, and where he's talking about Wisconsin. Now we are going all the way up to the frozen north in the Red River Valley, or the not so frozen north this year, but it's the same everywhere. And we are still talking about cover crops, but we have a little bit of a different system, often following spring wheat. Of course, we are in sugar beet country up here. So, but there are definitely some things that we're learning about sugar beets that are going to be applicable to other rotations and other crops too. So one of the things that has started happening, and it happens all across Minnesota, is people starting to experiment with these different conservation practices, strip till being one of them. So, when we have this project to look at strip till sugar beets, I thought it was a great opportunity to also start looking at then what's the effect on the nitrogen fertility, especially when we're in the colder and ever so slightly drier climate of Northwest Minnesota. The other piece of this is that beet farmers will grow nurse crops along with their young sugar beet seedlings. So sometimes they will plant oats at the beginning of the season. So you do have kind of an early season cover crop. And as comes to no surprise, especially if you listen to Matt's talk just before lunch, I mean, nitrogen cycling can be really hard to predict due to differences in the soil, weather, and management conditions. But we do know that in-season mineralization has an effect on our sugar per acre. So if anyone out there is not a beet grower, I mean, sugar per acre is really where the dollars come in. You want to have a beet that is very concentrated in sugar. So you don't want it to grow too big because then the sugar will be less concentrated. You actually want a big beet, but you want it to be high in sugar. And so we have seen where if you don't have enough nitrogen in the system, you then don't grow as big of a beet. If you have too much nitrogen, then your beet grows too big and that sugar's not as concentrated. The other thing that we see, and this is definitely more common in kind of the southern Min sugar beet region, sugar growing region, is that when people are following beets or following corn with beets, we are definitely seeing that there is some sort of tie-up happening, some sort of need for a nitrogen credit in those scenarios. So what we had here is we had a strip till trial that we were going to be putting in at the Northwest Research and Outreach Center. So I decided to monitor the nitrogen levels throughout the whole growing season. Now, one thing to keep in mind with this data is all of these plots were strip tilled into spring wheat. So you still had a lot of straw residue in the plots, and that doesn't really break down in the winter. So in the spring, you still have a lot of that straw residue. We did have fall or spring different seeded cover crop species, and again, kind of looking at that same sort of thing, if that decomposing cover crop residue ultimately affects sugar quality and yield. Now, this is just a picture of how the strips looked in the fall right after we put them out, and we did grow, we seeded the fall seeded oats, an oats and radish mixture, which had come up in the questions earlier about what happened to radish. Well, we did mix them with oats for this trial. Fall seeded winter rye, because we were pretty confident that was going to survive the winter. Hairy vetch, and a spring seeded oats. And as I mentioned before, that's the nurse crop. And if you probably can't see this, but if you squint really hard, there's like little teeny tiny cover crop residue just barely growing up in the fall for these strip till plots. Next, I'm going to kind of take you through what this actually looked like during the growing season. And what I want you to pay attention to here is to that residue. Here you can see we had a lot of volunteer wheat actually survive the winter in these plots. So there was cover crop biomass kind of throughout everything. It wasn't a very consistent plot, a plot by plot. It was patchy, but you can definitely see that wheat straw in these pictures. 15 days after planting, 16 days after planting, the sugar beet seedlings just barely starting to peak up. Then as we move forward, after we terminated these cover crops about 20 days after planting with glyphosate. So those Roundup terminated cover crops. And that did a pretty good job of knocking all that residue back. But 20 days after planting, that is getting pretty late. And that was a decision that we made due to some of the data we were collecting, not necessarily what we would recommend, but that's how it turned out this last year. And then as we move forward in the growing season, you can start to see that residue is really breaking down. It's kind of straw-like, but the beets are coming up. And then even as you move further in, about 46 days in the growing season, about a month after termination, a lot of that residue then has been broken down. It's laying flat on the ground. And this is about the time when the sugar beet canopy closes and we start to really see the beet plots looking like normal beet plots again, which is nice. And I actually don't have a picture of the beets after that, but you'll just kind of have to take my word for it that, the plots with the yellow flags in them behind the equipment is actually our plots. And they look like any other sugar beet plots, which is kind of why there are no pictures, is they stopped looking interesting after the canopy closed. But I kind of bring those through 65 days after planting. This is mid-July. Really being when, you start to see the canopy close, those beets have grown up, and then we harvest. So for this design, I mentioned we were tracking nitrogen throughout the growing season. So we looked at both ammonium and nitrate levels all throughout. About every 2 weeks. We went out and collected soil samples in these tubes. And this is going to tell us something about how the nitrogen cycle is moving. It's going to tell us how the microbes are using nitrogen, but then we're also able to see what's available throughout the whole season and hopefully see differences across the cover crop treatments. So this is a slide I use a lot. I've been using a lot, and it's because I don't think very many people have the nitrogen cycle memorized, and I wouldn't expect anyone to. I certainly don't have it memorized. Maybe I shouldn't say that with some of my faculty members in the audience, but I certainly don't have it memorized. I wouldn't expect you to either. So this is just your quick post lunch chemistry lesson for the day. And we basically have two forms of nitrogen. We have organic nitrogen and we have inorganic nitrogen. These are the two pools that exist in the soil in a broad sense of the term. Organic N, of course, we heard about manure earlier, and we're going to hear about manure again later. That counts as organic matter, but then also any residual biomass, that cover crop residue, also can contribute to this organic matter pool. To get from organic N to the inorganic N has to go through a process. There's a process called ammonification. And so when you think about, you apply your full rate of manure, but only half of the N is available, that's because it takes time for this conversion step to take place. Now you skip that step in the process anytime you add a synthetic fertilizer or a urea. You can just totally skip that whole ammonification step and get that nitrogen, hopefully, to your plants. Now you rely on the microbes in the soil to take that ammonium and nitrify it, it turns into nitrites and nitrates. And the nitrate is actually, that NO3 is actually what the plants can take up into their roots and hopefully turn into proteins or sugar in the case of beets, or really whatever you're harvesting. Now, that's not the only processes that happen. Obviously, we know that then that nitrate is also kind of that pesky nutrient that likes to leach. That's the form that leaches out. And it also can be denitrified. You can have other microbes. They can use that nitrogen to breathe. And so then they can release it in a process called denitrification, where it's released as either nitrous oxide or an inert N2 gas. And for this process, really what we're targeting with these in-soil measurements, we were measuring the ammonium, that NH4, and the nitrate, the NO3, all throughout the growing season anytime. So we would get a sense of what was in that organic matter pool, but then also have a sense of what directions these arrows are going. Because if you see a lot of nitrogen getting caught up in that ammonium phase or the ammonification phase, this cycle doesn't only move forward, it can also sometimes move a little bit backward through a process called immobilization, where microbes can actually use that ammonium for themselves and it doesn't convert all the way to nitrate. So for this talk, we're just going to keep it kind of simple, and we're going to look at just the nitrate. This is the soil test N that you would see on a soil test result anyway. This is, again, what the plant can take up, and this does give us a sense of what's available at any given time during the growing season. Okay, so this is the big graph of showing the different colored lines are the different treatments. We have days after planting across the x-axis and our pounds of soil nitrate on the y-axis. We also have a dashed line with the plots. That's our no cover crop treatment, but then the dashed vertical line that shows our termination, which again I mentioned was kind of like that 14 days after planting is when we terminated. And what we see here is that actually our treatments, we did see treatment differences between these different cover crops for most dates that we collected nitrate. We saw a lot of variability in the soil and what's happening. But one thing that's kind of important is that about this kind of 65, 70 days after planting, we did not see any differences by treatment. Now, this is this time when I mentioned that that canopy was kind of closing, the beets were putting on a lot of above ground biomass. And this ended up being one of the most critical points in the growing season that turned into recoverable sucrose. So we're seeing recoverable sucrose being really affected by how much nitrate's in the soil at this point in the growing season, but it's not related to treatment at all. So what's happening? We're seeing at this 64 days after planting, we saw that higher soil nitrate was correlated with lower sugar per acre. And that is kind of, it's a trend that you can see. You see that decreasing. As you get a lot of excess nitrogen in those fields, we had up to 120 pounds of nitrate left in the soil at the time when the canopy was closing. And we can definitely see that then our recoverable sugar per acre, our dollars were definitely down when we had that much nitrate left over. So I want to show you about what also, this amount of biomass is looking like. We know that we were seeing problems with nitrate. But I also think that that's not the only issue we were seeing here. We also saw a lot of biomass. And when Matt was talking, he was talking about, 1,000 pounds of biomass, 2,000 pounds of biomass. This is what 6,000 pounds of biomass looks like in a sugar beet plot. And you can definitely see we have other problems going on here besides just our nitrate levels. But what you do see here is, you see those plots being shaded out, but you can also see there's so much carbon left in these plots that needs to be broken down. And so what we're seeing when the plots look like this, in order to break down this residue, all of the microbes in the soil, they have to meet a carbon to nitrogen ratio to break this down. So in this case, we started to see that where you had a lot of residual biomass that microbes were tying up nitrogen. It was preventing it from making that final step in the nitrogen cycle to get to the nitrate step. Now on this graph, again, it's the same kind of plot with days after planting on the x-axis and pounds of soil nitrogen on the y. But the difference here is that the darker the color is, then the greater the biomass. So you start to see as the color gets darker, then you have more and more biomass, and you can definitely see dips at, soil nitrogen. 7 days after planting, you definitely had less, that 5,000 pounds of biomass right after planting, pretty soon after termination. It's tying up that nitrogen. And then we also saw more nitrate available at that 64 days after planting, at that time when the canopy is closing. And ultimately, we did see that then recoverable sucrose, instead of being necessarily correlated with the treatments we put out there, it was correlated with the amount of residue that was out there. So we really saw the effect of that excess carbon being broken down and it affecting our sugar yield at the end of the growing season. Because these treatments, we do see, nominally, when you look at the data, there is a treatment difference. But again, when you start to look at those other variables, like how much biomass and, what the soil moisture was and all of those other factors, you start to see it's not really due to our cover crop treatments at all. It's really due to the amount of biomass and just that variability that was present in the plots. So the main takeaway here is, again, we saw those differences in soil nitrate, but what type of cover crop you planted was not anywhere near as important as the amount of residue that was out on the field. And I think logically this makes sense and you're probably all sitting there and thinking like, well, this is obvious, right? But I think it's really important to then think about, especially in the context of what Matt talked about adding the nitrogen to the soil, adding that fertilizer to possibly overcome any of the tie-up that's happening. Residue management is really important. And we're also seeing this in other cropping systems too. We have another trial where we're planting green, we're planting soybeans into a growing rye biomass, and we're seeing something really similar, that this amount of biomass is really not a surprise. It's too much biomass, and it shades out the plots. It really causes a lot of problems, even for soybeans that are a little bit more resilient to those shifts in nitrate levels. And so I think one of the big morals of the story and what we're seeing pretty consistently is that terminating your cover crop after planting, 2 weeks after planting, is really probably too risky. It's even too risky when you're all the way up north in Crookston, Minnesota. So I think that really kind of puts things into perspective. If you start to see that rye heading out, go out and terminate it, and kind of keep that residue managed. But we will be repeating this trial in 2024. I like to think about, hopefully we'll be able to manage that residue a little bit better and get lower levels so that we can start to get a little bit more at the, amount of nitrogen that you might need to apply in a kind of more typical residue situation. We also are going to be adding a treatment to this trial, what's not a strip till treatment, so that we have something to compare to. That was something that I really wished I had when I started looking at this data, is like, yeah, this makes sense for strip till, but what about in that conventional tillage situation? So next year we are going to have a treatment that looks at that. But I don't want to leave you with the impression that strip till in sugar beets is impossible, because this is from a grower's field that's not too far from Crookston, and he's running a strip till machine, drilling the rye, and you can see how much better that residue is managed in his system, that he's been at it for a few years, than in ours. So you're still getting that rye growing up, keeping the soil in place. But here you can tell it's between the rows nicely. It's not competing with the sugar beets. So it is possible to do in a way that doesn't affect the crop, just not necessarily in the way that we did it at the research center this past year. So with that, just wanted to thank all the collaborators on this project, and to AFREC and to our Environment and Natural Resources Trust Fund that originally funded the trial. This is a picture of the strips going in this past fall. And I think with this warm weather, we're going to have some pretty good cover crop growth and see some different species, and maybe we'll see some different species effects next year. But with that, I will take questions. I know you guys got a lot of your cover crop questions out before lunch.
The picture that you just turned off, the farmer's field, was that corn or was that beets after wheat, or was that beets after small grain or after soybean?
Yeah, so the question, yeah, they probably heard the question because you used the mic, but that field, I believe, was also after a spring wheat. So they may have done a tillage pass. I'll have to remember, but even from looking at the field, you can tell there was not that straw residue left in the field like we had. They had a consistent surface that they then strip tilled into, which is different than what we had. So I'm not sure 100% off the top of my head, but yeah. But I think they did maybe do a tillage pass. One of the challenges with beets and spring wheat and trying to do those is there isn't a perfect offset of the rows. The rows don't really work as nicely together as, wheat and soybeans, or, even corn and soybeans. So sometimes that has to be worked out. Okay, the question just popped up. Am I familiar with the cover crop efforts in southern Minnesota for wind protection? Yeah, I am definitely familiar with that wind protection in southern Minnesota. And I say, like, this is really something that's of interest to us up in northwest Minnesota as well, because you just have in those flat plains, you have nothing to break up that wind. And so the wind can be really, really brutal. So I am kind of familiar with some of the cover crop efforts, and yeah, and I think that's what we're really hoping to see too, more so than anything else. We have a graduate student actually right now who's working on collecting some measurements in some of our cover cropped fields on how much sediment is moving in the wind over the winter. So again, I'm hoping for good results off of his project, but yeah, hopefully we'll have some actual numbers in the future. Okay, we track the carbon to nitrogen ratio on the cover crop treatments and how they influenced results? So that was actually something that we did not collect in the past year, but after I looked at the results, wished that I had. So I think it's something that we're definitely going to dig into for this next year, because that seems to really be affecting, the tie up of the nitrogen. And even as, Matt said earlier in his talk, I mean, there's a calculation you can do to kind of try to predict this. I think it's really important, but it was something that we didn't necessarily know to look for until we got out and collected data. Okay, this question, do you think there's a difference among sugar beet varieties response in this trial? I don't know. I don't know that anybody has really looked at, the differences in sugar beet varieties for this particular thing. So, I say I don't know, but I think that's an interesting question. Ooh, how would you get manure into soil on a no-till cover crop system? Ooh, this one's a hard one. I would definitely say, if you're injecting manure, I think this is where some of that wiggle room. I personally, some no-till purists would say, maybe not count injection. I certainly would count injection as a subsurface placement and not sort of violating that no-till principle. It's really, really important to get that manure in contact with the soil. I mean, you guys all know that with, the ban. If you listen to Melissa, you listen to other people's talk, if you leave the manure on the surface, it's just a lot more likely to slide off into the river. So getting it in contact through, subsurface injection, and there's a lot more technology about subsurface placement for dry fertilizers too than there used to be. That's really, really what I recommend for reduced tillage systems.